Serendipity brings us sometimes somewhere else than where we wanted to go originally. And the drug targets for oncology sometimes are very similar to drug targets for other diseases. As you'll see, we're opportunistic, and there was indication that we're not oncology, although we are focused more on oncology. And this was really helped by the creation of a program several years ago that was called CCR, or CSER, which was aimed at helping academics to translate discoveries into commercialization entities. And so we were fortunate enough to get funding from the federal government to the level of about $50 million over the years to create IRIC, which is IRIC in commercialization of research. The goal was to try to bridge the gap between the discovery and clinical assays, clinical trials, or partnership with the industry, or creation of company. So that's what we tried to put together. So both on the business side, commercialization side, but also on the discovery side of things. And part of the solution was, as I said, the creation of IRIC, which is the idea that this IRIC can invest in very well-selected projects that are very well assessed in terms of their potential scientifically, but also potential on the market, providing industry standard professional product management, quality control. As you know, one of the issues plaguing basic research is the lack of reproducibility in some of the findings which are reported. So it's very important to make sure that it's very high quality. Project management is also very important because there's a distinction between discovery for drug discovery and just free discovery, where you would go sideways rather than straight towards the target that you were aiming for. The privileged access to cutting-edge platform, that was very important. From the get-go at IRIC, there was a number of platforms that were built that were really centralized platforms, not in one person's lab, but really centralized platforms, top of the line, that were amenable for drug discovery activities. And then, of course, IRIC, over the years, developed track record. Were successful in partnership with pharmaceutical company, but also creating company through, in particular, out-licensing opportunities. The other portion of the solution was the creation of a drug discovery unit within the institute. So the institute is about 400 people total, including student, administration, everything. So it's 400 students, 400 people. Among these, 65 are fully dedicated to drug discovery program that originated either at the institute or in collaborators' lab. Among these 65 drug hunters, if you will, 50 of them are med chemists. So really have intense medicinal chemistry activities to bring the compound to the right level. Over the years, there was more than 45,000 new molecules synthesized at the institute. And right now, the Chimiothèque, which is now the Chimiothèque Nationale du Québec, CNQ, I'll say a few words about that, is roughly 130,000 compound. And now, this is a bit outdated, we have now 4 DEL libraries, DNA-encoded libraries, and totaling about 7 or 8 million compounds. And we had 5 drug candidates in clinical trial. All that generated through this team of drug discovery unit. The Chimiothèque Nationale du Québec now involves compounds from Université de Montréal, Université Laval, Université Sherbrooke, where people have put all the compounds together in a centralized chimiothèque, where everybody could have access to these molecules to run screening or to query the library. So I will just now give a few examples of what has been accomplished. I cannot give you the entire portfolio because it would take me much too long. I will just give you a few examples which have different flavors. So one of the, I think the recipe of success of the model was to be flexible and not have dogmatic views of what we should be embarking into. So one of the success that was achieved was through, and here, stick me, was something from my lab. So it was the putting together biosensors based on the technology that we developed many years ago, which is bioluminescence resonance energy transfer, which allowed us to monitor the signaling activity of hundreds of pathways in cells. And so this technology was clearly of use for drug screening, but not only screening, but profiling of drugs. And we were approached by a biotech company, a French biotech company, Domain Therapeutics, who wanted to license the technology. And we decided that it would be okay. They could license it to use it internally for the drug discovery program and also for their services, because we were also doing service for big pharmas. But for that, we asked them to start a site in Montreal. Otherwise, I would not license the technology to them, which led to the creation of about 15 jobs, which are still ongoing, and a lot of revenues from sub-licensing of the technology to big pharmas. And this is in the tens of millions of dollars of revenues that were generated. So each of the slides are designed the same way. For the three examples I will give you, on the left side are the scientific activities that occurred. And on the right-hand side, the business activities that were accompanying these different steps. And that is very important because we often wait until we get to what we think is close to the finish line before thinking about the business approaches that would be needed to get a success out of it. And in fact, these two things should get hand in hand in terms of progressing together, having a real plan to get a success out of these discoveries. The other example I will give is a example coming out from a collaboration that we had for many years with Bristol Myers Squibb. The collaboration lasted for more than 15 years. It's now reduced in size, but we're still collaborating with Bristol Myers Squibb. I'll give you one example that we brought in 3 programs, compounds into clinical trial. I will give you only one example. And again, on the left side is the discoveries, on the right side is it was the business side of things. And this is an interesting scientific story because this was targeting PAR4, which is a G protein coupled receptor, protease activated receptor type 4, for stroke. And at the time, all of the KOL were saying, no, PAR4 is not the target. It is PAR1 that is the target. And one of the reasons for that is that there was no good animal model for PAR4 because it's not expressed in rats, in mouse, but we knew it was expressed in guinea pigs. So we had a model. And so we took it with Bristol Myers Squibb all the way to a successful phase 2, a couple of successful phase 2, and now it's going to be entering phase 3 in 2025 for stroke. So I think that's a very good example of the success of a molecule that was made here in Montreal and has been licensed in a sense to Bristol Myers Squibb, although this was a really partnership all the way from the beginning to the end. Just to mention for the students that you don't have to sacrifice publication because you're doing a partnership with industry. Of course, you cannot do it at the same pace. You cannot do it at the time you want to do it, but we've published a lot of high-level quality paper, high-impact papers with Bristol Myers Squibb based on this project. And here are some of the people who contributed to the program at Université Montréal, Madeleine Roux, Mario Caleggio, and Anne Marigny, who is the director of the drug discovery unit, a medicinal chemist. The last example I will give is one coming from the lab of Guy Sauvageau, where they were targeting leukemia and led to the creation of a spin-off company known as Exceltera. In that case, this is sort of a funny story because they were looking for something that would be expanding hematopoietic stem cells from blood cord for transplanted leukemic patients. And so they screened phenotypically for compounds using our collection of compounds. The molecule that came out as active early on with a couple of micromolar affinity and not very high was a molecule that was part of another program at the Institute on a GPCR actually. Nothing to do with it. And then from there, they were able to move it forward to create UM171 that is now has a generic name, which is DRUGGABLE, which is now awaiting the authorization from EMA. They had the nomination for accelerated declaration in Europe, and also they have submitted the documents at the FDA, so we're hopeful that this will happen. But already in the phase 2, they were able to treat over 100 patients with incredibly good success in the the outcome of the patients. So just to give you an idea of the amplitude of this, over the years, the Institute has 20 years, but IRIC has 15. So let's say 15 years. We have structured and financed over 100 drug discovery projects. There was more than 50 public and private partnerships that were established. 30 licenses were made to the industry. There was more than 326 patents in 60 different families, and more than 150 were submitted. As I said, there was 5 new chemical entities in 5 clinical trials. We started 6 companies, and 5 are still in operation. And we also supported the people that wanted to do entrepreneurship, internship in terms of learning how to become an entrepreneur. And finally, over the years, more than $150 million were generated in R&D and revenue, with potential of $2 billion in license revenues. Of course, if everything was going perfectly, it's not gonna happen. We all know the success rate for drug discovery is not that high, but just to give you an idea of the amplitude of the programs that were done. 30 seconds is a very good thing because this is my last slide. So thanks.